We're going to begin. I'd like to welcome Carlos to give a talk today. And of course, I don't have his talk in front of me, the title of it. I know all about what it's about, but of course, I don't have it sitting in front of me, but you guys all do. But we're excited to have him talk about how do we deliver precision health at scale for all. And Carlos is back at Stanford. I'm delighted to say, is still in part running his company, but will be back, I think, full-time next month at Stanford, and has, as you know, a former co-chair and active member, former PI, co-PI with Sharon for the Clinton grant at Stanford that I took over for him. And he was a co-chair on the ADWG and has, with all of other things going on, stepped down from that, but has been an active participant in ADWG along with complex disease, HLA, et cetera. So Carlos, I'm thrilled that you accepted the invitation, and we'll turn this over to you.
Thank you so much, Terry. It's a real privilege and pleasure to present to this group. First, am I showing the right version of the slides, or do I need to flip?
No, it looks good.
Okay, great. Yeah. And so, as Terry mentioned, this is a combined deck that largely focuses on a project that we started at Stanford, and I want to begin by really acknowledging the extraordinary work of my colleagues on this project, and then where we've taken part of it in the private sector to really build a public-private partnership that we're calling the Biobank of the Americas, and hopefully, if time permits, to link it back to some very interesting biology that started me off on this whole cascade of thinking about the long-term consequences of infection. So I'll begin by acknowledging you and Ashley and Vicky Parekh, who really collected a lot of the genetic data that I'll be talking about, Manny Rivas and Alex and Arturo, who've been, we've been kind of like the Beatles on the data analytics side. Jenny Frankovich, who's my long-term collaborator on post-infectious sequela, particularly in children. And then the real star of the show here, who is the unsung hero, is Dr. Ben Pinsky. Ben runs viral testing at Stanford, and nothing that I talk about today would have been feasible without Ben's just, unbelievable extra human effort over the last few years to collect all of the COVID material that became the centerpiece of what I'll talk about. So I want to frame it in really a way that many of us already like to think about the value of genetic data, which is as a way to ultimately de-risk how we bring new drugs to market, right? And I like to say, to me, PCSK9 inhibitors are the best example we have of a bench to bedside logic that has now been turned into almost a factory of new discoveries that are at different stages of prosecution. The best example is probably the monumental effort of the UK Biobank, where 8 pharmaceutical companies financed the accumulation of data on 500,000 samples that many of us rely on every day to do population genetics, to do medical genomics, to do drug discovery, to do target validation, to do hypothesis testing. I mean, there is no resource like the UK Biobank. And even in 500,000 people, you find enough signal to at least get 4, if not more, molecules to different stages of development. So it's just an extraordinary resource, and I love the UK Biobank. The biggest problem, of course, is that it is not representative in any way, shape, or form of most of the global diversity that we'd like to be able to ultimately impact. And so we began calling attention to this problem more than a decade ago, along with many others. Unfortunately, the statistics have just not gotten better over time. So even as we've become kind of a thorn in the side of teams that build these kinds of resources, the train has been a runaway train. And I want to just, point to one very nice paper by my former student, Alicia Martin, who then was a postdoc with Mark Daly and is now an investigator at Harvard. Sharon and I just saw her last week, 2 weeks ago. She was a panelist on Precision Health Summit at Harvard. And these are statistics that summarize the number of individuals in GWAS studies over time. And again, you don't need to be a statistician to see that the red has continued to grow exponentially. There's a little bit more blue, which is East Asian representation, largely from the very logical and involvement of China in genomics and investment in genomics. Even if we don't have access to a lot of that data, it does exist, and there are publications out there. Unfortunately, the rest of the world has not been anywhere near as represented. I would even argue within Europe, it has largely been the UK, the Finns, the Danes, maybe the Dutch, and certainly the Icelanders. Anything sort of south of France, you also begin to see underrepresentation in, and anything east of Berlin, you see underrepresentation in. So it's a very small pocket of Northern European ancestry that really forms the basis of everything we think we know. And this has real implications, right? Like, it's not just a theoretical harm, it's an actual harm. The implementation of polygenic risk scores do not work as well in African, East Asian, South Asian, or US indigenous and Latin American populations as they do in Europeans. And so it's a real harm. You really are creating health disparities simply by letting the world evolve as it has for the last 20 years. So what are we trying to do? And we're trying to float on what I think is an inevitable trend, that of digitizing electronic health records. To me, this is obvious. It's like sitting in a boardroom in the late 80s, early 90s at a telecom and asking, I wonder if they're going to want cell phones outside of New York City and LA. Yeah, they're going to want cell phones outside of New York City and LA. Like, think about the middle of the country, think about other countries. They're going to want cell phones in Mexico. They're going to want cell phones in Colombia. They're going to want cell phones in Cairo. They're going to want cell phones in South Africa. So it's obvious to me that the digital health records are, just going to get more and more of the norm. And, COVID has accelerated this to some degree. And the big question is, how do you get samples from underrepresented groups in a way that really can scale and engages in a way that has the ability for that material to have at least UK Biobank-like reach through. Ultimately, the other piece here is that this isn't just the right thing to do because you're bringing genomics to understudied populations, but it's the logical thing to do. Even if you just literally care about bringing drugs to market in Europe, the data outside of Europe is going to help you better adjudicate your target. So even if you're an incredibly just, focused individual on what pharma to bring, this helps you. Right, so there really is there's a Pareto optimum. You make everybody better off without making anybody worse off. So why Latin America? Well, to me, it's obvious just because I happen to be from this corner of the world, and we're set up here in Miami. But Latin America is a wonderfully diverse place. It is 10 times the size of the UK. So there's 65 million people in England. There's 650 million people in Latin America. There are at least 700 ethnic groups, 33 countries. If you like founder populations, Latin America is full of them. If you liked Decode because it was an island, and then Latin America is full of them. Cuba's an island, Hispaniola is an island. The valleys in Colombia each create islands. So it is a geneticist playground. In fact, it is ironic that it is underrepresented, as the first place we ever got to a gene for a trait of medical significance was the Huntington's disease gene that got mapped in a village outside of Lake Maracaibo, a few hundred miles from where I was born in Caracas, because the village is one of the highest incidents of Huntington's disease, you phenotype by eye, right? So it has always been of interest to geneticists. It has now reached the level of sophistication, as if we want to use that term, or complexity, that a lot of the data is also digitized. So there's really no reason not to engage now. So our thesis is that much like in the same way you can build cohorts outside of Latin America, you can build them in Latin America and in Latin American diaspora populations, and that the public is willing to give you a sample in exchange for insights about their genome. And this is actually not a crazy hypothesis. This is actually held and consistently across surveys from many parts of the world. Somewhere between 60 to 70% of people are willing to take a DNA test, even excited to take a DNA test if they're getting something of value back. It just happens to be that not everyone has $200 to send to Mountain View or to San Francisco to get a 23andMe or ancestry test, right? And, they've got other things to do with their money. And so how do you make this available to a broader swath of people, right? So at free, how many people would take a DNA test? About 60 to 70%. And you can actually, as of 2017, see how the direct-to-consumer genetics market began to tap out, because by then, people making over $200,000, 60 to 70% had already taken a DNA test. Most of that was actually gifted DNA tests, not something they had necessarily bought for themselves. Yet, these are US data, yet less than 10% of people making under $15,000 a year had taken a DNA test, but 60 to 70% of them were willing to do so, right? There's a 20, 30% of them, of the population, who's not interested in taking a DNA test. They don't know why they'd want a DNA test. They're weirded out about it. Great. 60 to 70% is more than we need for what we're trying to accomplish. So what are we trying to do? We're trying to build a biobank of consented samples. We're trying to build world-class analytics, and then create a federated discovery engine that can be self-sustaining, right? That's the goal. And what I want to tell you about is why we began specifically with COVID as a source material, and how the Stanford experience can actually be replicated. So I'll talk a little bit about what we did at Stanford. We are, of course, nowhere near the only people who thought about doing GWAS from COVID material. In fact, almost every major company that had access to material got involved in contributing to COVID genetics efforts that included common variants, rare variants. We were part of the work with John Laurent Casanova. That this is a drop in the ginormous bucket of what we have learned about all kinds of conditions, including phenotypic, just typical phenotypic variation across humans based on a post-COVID environment. And as an evolutionary biologist, this is just canalized variation. These traits did not exist until COVID unmasked them, but this is exactly what you expect from evolutionary first principles, right? There's hidden genetic variation in any population, and once you shift the environment, you get that revealed, be that heat shock or COVID, which is a form of heat shock, if we want to think about it that way. So this is the building I'm in right now. We're in a 10,000 square foot warehouse in Hialeah, Florida, which is the industrial heart of the city. 250,000 Cuban expatriates call this home. It is the La Ciudad que Progresa, the city that progresses. Across the street is the Jose Marti Child Care Center, and it is my little piece of heaven on earth. We stood up free antigen testing for the community, and we're a suspicious bunch, and so not everybody signed up, but you could get a reasonable 10 to 20 people to and give you a sample and sign a consent to be part of a 10 million person IRB study. We then scaled this out, working with real professionals. So there's a group called BlueMed that was doing a bunch of testing here, and then now we're working with a group called Nomi Health that has the contract from the Madam Mayor of Miami for Dade County We're excited to work. At any one of their sites, they can get 30 or 40 samples a day. And then if you just sort of network the sites, you can imagine you can get hundreds of samples a day. So I'm going to show you some initial data on individuals we were able to study here as part of this process. This is real live data from Miami, where in blue here are the patient samples that we recruited. And then in the rest of the PCA is colored according to the kind of traditional 1,000 genome ancestry grouping. So here's the corner of European diversity space, corner of African diversity space, corner of Indigenous American and East Asian diversity space on the third and fourth principal component. So when all is said and done, we do an incredibly nice job of covering that multidimensional space. And if you want a kind of apples to apple comparison, here's the UK Biobank, 95% European, less than a fifth of a percent Hispanic, 1.5% African. We are the Pareto opposite. We are 20% European, 80% non-European samples, with Hispanic Latinos making up 33%, African American 30%, and Asian, in our case, South Asian 14%. We can talk a lot about why there are South Asian ancestral groups in the Caribbean. It of course has a lot to do with the colonial history of the Caribbean. So it works in the wild. You can get people to recruit, and we're building now this resource called the Biobank of the Americas, where you too could participate were you to choose to do so, and get in line to get a potentially free genetic test once we have sufficient resources to get to everybody. Certain groups will get prioritized based on both a presentation and what we call the automatic chart review for people who are willing to share their clinical data with us. So here's how we've fared in the last year. We literally started this about 13 months ago, and we currently have 100,000 samples in the bank. So I have a fifth of a UK Biobank sitting in commercial grade freezers. Kind of see some of this behind me here. When I say a warehousing district, I mean a warehousing district. And then
Carlos, you've got at least one hand up. Do you want to go through your slides and then take questions, or are you okay to be interrupted?
Go ahead and interrupt.
Claudia, you had a question?
Yes, thank you. So I'm just curious as to how much do the participants know and understand that by signing up to this biobank, like they are also consenting for their samples. It's not just about the free testing that they're getting, but that they are consenting to other research and for-profit research, and sort of eventually maybe developing drugs based on this information that they are donating. Sure.
Yeah, we have a 10-page consent. We go through that consent with the participant. Again, not everybody signs up. If I told you I had 99% sign-up rate, I would expect there'd be some eyebrows. It's closer to 50 to 60%. And then we also have individuals who change their mind and pull out of the study. So we are actually pretty comfortable with that. This is a approved study both by Western IRB, which oversees this kind of commercial type recruitment, and we've also gotten through university partnerships. So Miami-Dade Community College, which is the big engine of social mobility here in South Florida, 150,000 students, just approved us to be one of the only commercial and non-commercial sites that's able to recruit on campus. So we've stuck ourselves with a very high ethics bar. This is not sort of pass-through testing where you go do your COVID testing at the university. You don't know what's happening to your sample. We actually ask people to enroll, and they are enrolling initially in a de-identified database, and then we also ask them to be enrolled, be contacted. So all good questions. And in fact, the one overwhelming question we get is, but the government isn't getting my sample, right? Nope, we're under no obligation to share your sample with the federal government. Great, then sign me up. So in fact, there is often, and this is just the reality, people understand that their data is being used by companies all the time. We all understand that when you say something in Siri, now reprocesses it, and the next ad you see is for whatever you were talking about, right? That it is part of what happens with data. What they don't want is that information going in the hands of immigration. They don't want it going in the hands of FBI. They don't want it going in the hands of even NIH. Many people do not trust the NIH, but they're happy to trust a local laboratory that set up shop right here in Miami in order to work on exactly this problem. And most of them donate for the reason that it will benefit the community. That's the number one reason. Is this data going to be used to help Hispanics? We hope so. That's the goal. So it's an important question, and it's one that we try to take straight on.
So, sorry, just a follow-up question, if I may. So
Sure.
to your this last point that you just mentioned in terms of people signing up to contribute to better understand their community and that improvement of the community. So how much of that is really part of what you are either currently or eventually planning to do in terms of, I don't know, return of results or community
So
engagement?
I think that's probably a broader question that maybe we should save to the end, because I think we'll probably hear
Sure.
some of that.
Yeah,
Sure.
no, I'll quickly answer that. We've created an internship program for Miami-Dade Community College students to help us with exactly these issues. So they both come in, they learn to biobank, they learn to process samples. There's a massive need for licensed technicians. The state of Florida is actually second only to New York in terms of the complexity of getting clinical genetic testing up and going. And so we want to create a path towards clinical genetic expertise in the community, which is why we have a dozen Miami-Dade interns, day in and day out working with us, and many of them are integral to our recruitment. So we have Haitian American students, we have Haitian students, we have Cuban students, we have Colombian students. Every campus is a little bit different. West Campus is more Venezuelan. The Hialeah campus is more Cuban, but they all want to be part of it. And in particular, this is a very family-based town. So if you're recruiting at Miami-Dade Community College, you're often getting not only the kiddo, but you're getting mom and dad and abuela and tia, and they're all concerned about the medications that they're on. And so they're very interested in pharmacogenetics, for example, and they're very interested in HLA as part of the things that we're focusing our efforts on. But great questions, Claudia. The other thing we do is build infrastructure on the analytics side so that data doesn't need to travel, but models can. So we are also operating ex-US. Oftentimes clinical data can't leave a country, or even genetic data can't leave a country, but we can generate models and update models with the partial likelihoods traveling as opposed to the samples or data traveling. And that's an area that we're very excited about, and one that builds on expertise in biomedical data science that comes from the Stanford side. We're also part of the global bio as well, and our goal is that are scaling out this kind of effort, and we want to make sure that our data is interoperable. That's a huge piece of what we want to accomplish. So I want to talk a little bit in more detail about the Stanford experiment. So this was now been published in Nature Communications, where we took the primary swab that was being used to test and looked at the first phase of the pandemic. We were able to get low-pass whole genome sequence data, HLA typing, viral genome data, and host transcriptome data. We focused on 2,000 COVID swabs out of by then 250,000. There are actually more than 1 million swabs that Stanford passaged in the pandemic, and this is part of why Ben Pinsky is a personal hero of mine. I want to spend a little bit of time on the next two figures that really demonstrate some of the fascinating patterns that we saw in the data. So this is a summary of a structure-like plot where every individual is represented as a bar in a bar chart with ancestry components that again mirror sort of the canonical 1,000 genomes reference population. So these are individuals of African American ancestry. These are individuals of East Asian and sometimes East Asian Oceanian admixed ancestry, individuals of European ancestry. These are mostly individuals with high indigenous American ancestry sampled in the Bay Area, and individuals of South Asian ancestry. And what you can do here is compare the COVID positive patients with time matched COVID negative patient controls. So again, you don't need to be a statistician even to see there's a lot more green in the top and little bits of pink relative to the bottom. And so there are real, disparities, as it were, between who ended up in the database as a COVID positive patient versus a COVID negative patient. This is literally the very first phase of the pandemic. This is pre-vaccines. This is pre-Paxlovid. This is, really the first wave. We also built a model that included relatedness among the viral strains. This is all alpha. This is even before you get delta. Severity score, and demonstrated that this wasn't just community level transmission. Rather, there are clusters of cases that kind of co-travel together. But my favorite figure is this one. So this is self-reported Hispanic versus non-Hispanic over time. And in the bottom are the exact same bar charts, but now stacked, that give you a sense of the volume. So from March to early April, this looks like very typical Stanford patients, right? Mostly European, some East Asian individuals, a lot of house staff. As you go into April, May, and June, you begin to see far more Indigenous American chromosome and African chromosome representation in the database, as well as an overwhelming signal of HLA association, including A31. A31 is actually a known pharmacogenetic associated allele, right? And so this, of course, has everything to do with social determinants of health. Who can be a DoorDasher and who can't be a DoorDasher? These are mostly, mixtecos from the Central Valley that didn't, have the luxury of, Google letting you work from home, right? So we want to disentangle the social determinants of health from the potential for ancestral allelic differences that through no fault of anybody predispose or don't predispose you to COVID, right, and COVID severity. And the way to do this is to rely on a very battle-tested approach, which is admixture mapping. So if we think back to the early days of genetic mapping, admixture mapping for prostate cancer, it made use of the fact that if you were to look at the epidemiology, African-American men present with higher incidence of prostate cancer, but in terms of their genetic ancestry, that should be randomized across the genome. So if you look for segments of the genome that are enriched for ancestry, they may overlie true ancestrally differentiated alleles that again are just the random lottery of the genetic drift. And so this is an algorithm that Alex Ioannidis pioneered. It's called genomics. It is a sort of best in class way to do this local ancestry deconvolution. As they say, a picture's worth 1,000 words. So one of the big challenges in doing this kind of work is that if you just run a classifier, it tends to over call ancestry switches, right? Because it's very easy just to pick what's the best ancestry at a given location. And so the key is to add a smoother that allows you to account for the limited number of generations back to the time of admixture, and then put that in as a way to really improve the resolution. When this is all said and done, you can compare it to a fully generative model. Again, this is a really shout out to Alex who built these wonderful tools and actually won some competitions at the AI and deep learning and community meetings. These are fully, generative models that allow you to also generate synthetic admixed individuals that allow you to check for local biases and how you deconstruct ancestry.
So
again-
Carlos, can I interrupt for just a second? Or
Yeah.
when we get a chance, can you stop so we can, you've got some questions and a couple hands up.
I will be done in 2 slides,
Got it.
at least with a logical place to break. So when
Perfect.
all is said and done, you can actually go through and find peaks of actually African ancestry and Oceanian ancestry that meet genome-wide significance and are associated with susceptibility alleles. So we were excited to see this, even though a lot of the initial signal came from individuals of indigenous American ancestry. The true admixture signals actually come from differences in African versus Oceanian ancestry. The other one that's very interesting is a protective HLA allele in South Asian populations that, from a modeling point of view, actually explains a very quixotic observation, which was that there was no COVID in the Indian subcontinent until the summer, or there was very little COVID in the Indian subcontinent until the summer. And that's when Delta broke through. So when you do the epitope modeling, it looks like that may be the cause. Okay, with that, this is a natural place to stop. There's additional things that we could talk about in translating this into PANS, pandas, and other conditions, but happy to take questions that folks may have.
Great. Thank you, Carlos. So let's start with Jen. She had her hand up first, I think.
Well, yeah, and sorry, because you just introduced all that interesting admixture stuff, and I'd love to hear more about that. But my question dates back to the previous idea of consents and discussion. And actually, Alice has asked the same question in the chat, which is thinking through this, people not trusting their government and not trusting NIH. What does that mean for NIH funded research and our data sharing requirements? And how do we work through including the diversity while dealing with that mistrust, I guess is the best way to ask that.
Yeah, I mean, look, the consent is a conversation. It's not an action, right? So you can always recontact people and say, hey, we now have an opportunity for your data to be individually enrolled in a study. Would you choose to do so? Can you opt into that? And there's some folks that would be willing to do that. There are also summary levels of the data that can, of course, be shared, right? Someone's HLA type is not identifiable. And so we can always share frequency data, not unlike other consortia. So, our goal was to build something that was flexible, that's a conversation with our participants, in the same way that to get access to the UK Biobank data, you need to propose a study, and then that study gets reviewed, and we, like others, have access to the data, right? So this is I think about flexibility and agility. Unfortunately, the government's a big bureaucracy. We all know that, right? And that's not a function of anything but the size of the engaged org, right? Everybody, in some sense or another, is government's the name to what we choose to do together, right? So we're all in some part, of the government, right? And so if the government decides that they want to incentivize data sharing, they can come up with ways to make that happen, right? In fact, the way that I understand it, that all of us was able to get their recruitment going is to work with federally qualified health services, right? We too work with them, right? You can be enrolled into multiple non-colliding protocols. So it's really all a question of where practicality meets, the regulatory hurdle. I would say my biggest lesson in this whole thing was don't be afraid of the boogeyman, right? Everybody says, oh, minority populations are really difficult. They don't want to be engaged. They don't care. Really? literally set up a table, go talk to people at the mall, right? Because we could do it. If we could do it, and we're, you know, a band of, you know, a couple dozen people right here in Hialeah, Florida, where my internet is so bad, I actually had to put a satellite on on my roof, right? I couldn't get Comcast to bring me fiber optic, right? I dial a modem. I'm using, you know, Starlink, right? So at the same time, there are many things about the local government that work great. We could not do this if it wasn't for the full-throated support of the mayor's office. The mayor, I met with both mayors of Miami. The president of Miami-Dade Community College was the first person to come and visit us. We've had the state representatives come and visit us, right? So there's overwhelming support, and you know, the reality is that, you know, there lots of ways that we can, you know, create partnership opportunities in the same way ClinGen, you know, in some sense got clinical laboratories to give up a bunch of their data, right? That would have seemed verboten, but you know what? We asked and we said everyone will benefit if we share data because it will help us adjudicate the quality of clinical genetic testing, right? You make the right value proposition. So what we can't do is basically say, I'm here from the government, give me your sample. That's just never going to work, right? That's always going to be kind of a bullying strategy. And so if you're willing to engage with the community, you got to understand there's a lot of different voices in the community, and those are conversations to be had. So, but one of the other interesting conversations we've had are with the migrant worker communities out here. They're of course very reticent to participate because they're at real, you know, danger of you know, deportation. They're a danger, all kinds of dangers, right? They don't even go to the free health service outside of the migrant worker camp, right? So if you don't understand those issues, you're not going to be able to navigate this successfully. And oftentimes what happens is that we tend to fund the elite institutions, right? So, you know, University of Miami, an institution I have a lot of admiration for and who, you know, worked with in the past, also recruited me for a COVID study at a local fruit shake place. It took them, you know, half an hour to have a conversation with me. There were 7 people from I got a $50 gift card, and then they couldn't return results to me based on the protocol that got approved by the University of Miami, right? So, you know, it all depends on what you're trying to optimize for.
Thanks. Bob?
Yeah, hi, Carlos. This was really interesting. I have a policy question for you,
Yeah.
and I'm wondering whether Galatea has taken any stand or has been interested in the issue of Senator Tillis's so-called patent reform bill, which is essentially designed not only to reverse Mayo v. Prometheus and also the Myriad, but is going to allow companies to patent relationships between variants and disease phenotypes, which to me, I think is going to strike at the heart of being able to use the sort of data you're generating to do drug discovery, to develop screening or diagnostic PRS scores in exactly this population, which is already underserved.
That, you know, that's a great question. To be honest, we've been heads down building and not. You know, we have no lobbyists in Washington. We have no lobbyists in Tallahassee, and we don't have a lobbyist anywhere. So we've not been particularly active on this issue. We're focused on, you know, operating within the current law of the land, and that in Florida also has requirements around, you know, if you've got a genetic discard, you got to tell people what you're going to do with it, right? So I do think that in general, patent protection has to be, you know, carefully balanced with what you're trying to achieve. So even if you think about the Myriad patent decision, it weirdly excluded RNA because RNA could be synthesized, and so, you know, and you go back on it, and there was like a friend of the court brief from the Broad that had IP around RNA, and you know, you kind of understand why the law supposes from the beginning that people have competing interests. That's the whole goal, right? For it to adjudicate between people who have competing interests. So nobody has, really no interest. And as they say, no conflict, no interest. So I haven't, personally delved into it. I respect your view, though, that anything that hinders research and open exchange of information down the road is probably going to be bad for innovation. The whole point of patents is that you tell people what you're doing, and then you can put it out there, and you have a protection. It actually goes back to the Greeks and the recipes for big feasts, right? So that literally people could know what's in the secret sauce, and then you're the only one that can make that secret sauce for a year or two. I think
I think because this is a ClinGen sponsored call, we probably shouldn't have a detailed discussion about pending legislation. Is all I was gonna say.
No, exactly. That's so. I'm happy to skirt on that and have a separate discussion. I think what's probably most relevant to this group is that I think we've crossed that threshold well before of, you're not going to build one database to rule them all, right? You're going to have to figure out how you get pieces of data to travel that you can then curate and make accessible in reusable ways, right? So one of our big challenges, I would say, at ClinGen was really getting over the threshold of trying to build an individual level database, and I think that's getting to be an even more fragmented challenge. But what's been wonderful is to be able to share data at the variant level data, and to bring in power of automated literature review and other things, right? So if you can circumscribe the project well, then you can build tools, because ultimately I think that's the most important thing to do. So I just wanted to close with a little bit of where this is going. We had a wonderful opportunity to work with a partner that had already collected a bunch of genotype data and was deploying surveys. We were able to validate using that resource cardiometabolic polygenic risk scores. This is a comparison of both all individuals in the data set and then only individuals of European ancestry in the data set. And so we came up with ways to bring the admixture element into the equation. We can discuss till the cows come home of how polygenic risk scores are going to get implemented in the clinic and which ones and how and blah, 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 blah. But ultimately, you have to be able to validate these across populations and across heterogeneous data sources. And that to us was an important problem that we chose to spend a lot of time on. And I'll just close with this example, which is on the COVID genetic side. We also looked at a series of polygenic risk scores that combine these signals, and this is the slide from Manny Rivas. This was based on the UK Biobank data. And what became clear is that not only do individual variants or haplotypes predispose, that there were actually kind of entire elements of genome-phenospace, particularly around coagulation and deep vein thrombosis, that many others have also been able to demonstrate, and lots of ways that you could measure this in terms of ICD-9, ICD-10, lab values, diagnosed by a doctor, et cetera. But I do think this is where the future goes, right? Like there are layers of data exchange, and you're not going to ever want to move the raw image files from the NextSeq. Why would you, right? You're rarely ever want to really even share the FASTQ files. Why would you? VCFs are getting processed, so maybe you merge and share VCF files as you need to, but ultimately all of these derived analytics are where the knowledge base goes. And from again, just thinking about getting new drugs to market, it was the very fact that the genes that predispose to high cholesterol also predispose to early heart attacks, and cardiovascular disease that convinced us of the LDL CAD axis, and that the genetics of lowering LDL lowered risk that ultimately got proven out in that PCSK9 example that I showed. So if we think about that broader picture, if we're able to get to the outer regions of genopheno, find where predictions are made, reliably make predictions, and then find people who go in the opposite direction, right? It could have been that loss of function mutations in PCSK9 actually made the whole thing worse. It could have turned out that way. Helen Hobbs, who deserves the Nobel Prize, in my opinion, I'll go out and say that, the second Nobel Prize for the cholesterol pathway, Esther Brown and Golding. I'd give to Helen and colleagues on this. Is going to save millions of lives with initially PCSK9 inhibitors, and then if SEC and VERV are successful with, even vaccinations against heart attacks, if you want to think about it that way. And that's just one pathway that leads to one condition. Think about the totality of what we're learning as we digitize phenotypes, as we digitize health record data, as we move to scale out the ability to bring little genetic phenotypic signals to align, and those streams, go into rivers and rivers into oceans. So I'll stop there. Happy to stay on.
You got another question from Claudia.
Hey, Claudia.
Hi again. Just a very quick question. Since obviously you mentioned all the great characteristics in terms of genetics that the American continent has, and other populations in the continent that haven't been yet studied, and part of what you are intending is to build this American Biobank enterprise. So I'm just wondering, how is whether you have started or are planning to, but how are you going to engage with communities and researchers and populations in other countries? Because right now you're still local, right?
No, I didn't talk about, we have operations in Colombia, Peru, Paraguay. We're super engaged, much in the same way we did 1,000 Genomes, right? So 1,000 Genomes was the coalition of the willing that sampled Puerto Rico, Colombia, Peru, and brought in map 3 samples Barbados. So we are actively involved in international efforts. We fund universities and private efforts in other countries to get samples. We're planning to work with Tec de Monterrey and their Origen project. So again, it's easier for us to work with private organizations and directly with universities, and work within the legal confines of what data and samples can and can't leave a country.
One last real quick, Daphne, do you want to raise your question that you put in the chat?
Sure, and sorry that I'm gonna have to run in a minute, but I'm still not quite understanding the consenting process. Are people recognizing that there's a distinction between how their COVID test results might be used and who has access to that versus their sequencing data? I'm just not clear if people are being told that their COVID test results won't be accessed by federal entities, but they're in the future.
I apologize. You are obligated, if you're testing for COVID and you find a COVID positive, to tell the state authorities, right? Like that, and that's in the testing. So they get their paperwork for COVID testing. After they got their COVID testing, which is usually it's a separate organization. We're not doing the COVID testing. We're getting somebody else that's doing the COVID testing. We say, by the way, would you be interested in enrolling in the Biobank of the Americas? They actually give a separate sample. So
is their enrollment in the biobank, are they being told that federal entities will not have access to that data?
So you're giving a sample to they're separate samples. There's a COVID sample that goes through testing and then is destroyed. And then in order to make sure people aren't confused, we take an actual buccal swab for the research sample.
Right. I guess you had mentioned earlier that part of the reason people are putting their trust into you is because you're communicating that that the government is not going to have access to their data?
The question we get asked most often is our sample going in a federal database?
Okay. But
That's
but the data there will be data sharing with federal entities, with the genomic sequencing data specifically.
We will share allelic frequency that is consistent with not breaking anybody's identifiability.
Okay, so just to clarify, people feel comfortable, they don't feel comfortable with their samples going into a federal database, Biobank, but they do feel comfortable with their data being accessed by federally funded researchers to do genomic sequencing research or genomic.
Yeah, look, again, I think it's a great question. I would be very interested in even deploying a survey to our participants to actually address this. I gave an anecdote about the #1 question we get, and it's clearly raised some interest, and it would be great, Daphne, if you're interested, we could, if you have a survey in mind that you'd like to ask, we're happy to deploy it and see what people say.
Okay, yeah, I'll follow up via email. I think I'm still not quite following, but I know we're over time, so thank you.
Great.
Great. Carlos, thank you very much for giving the forum this morning. And welcome back to Stanford.
Appreciate it. Thank you, guys. Bye.
Thank you, everyone.